Bread, rice, and potatoes are dietary staples enjoyed across civilizations, yet their high carbohydrate density frequently raises concerns about postprandial blood sugar surges. To understand how glucose enters circulation, we must examine the biology of starch digestion. Dietary starch is composed of linear amylose and branched amylopectin polymers. In easily digestible or refined starchy foods, salivary and pancreatid alpha amylase, followed by brush border enzymes in the small intestine, efficiently break down these carbohydrate chains into glucose. Absorbed across the intestinal epithelium, primarily via sodium-glucose cotransporter 1 and related transport pathways, this glucose enters the bloodstream, contributing to the post-meal rise in blood sugar and prompting insulin secretion from pancreatic beta cells. However, clinical nutrition research demonstrates that carbohydrates do not behave identically in the body. The physical architecture of the food, molecular structure, and digestive context fundamentally shape the postprandial glucose curve.
One of the most widely studied food science strategies involves a physical transformation known as starch retrogradation. When starchy foods like rice, potatoes, or pasta are cooked in water, heat and moisture disrupt their crystalline granules, gelatinizing the starch and making it readily accessible to digestive enzymes. However, when these cooked starches are subsequently cooled, the linear amylose chains realign and recrystallize into tightly packed structures, forming type 3 resistant starch. Human studies suggest that cooking and cooling certain starchy foods can increase resistant starch and, in some cases, reduce the postprandial glucose and insulin response compared with freshly cooked versions. Furthermore, upon reaching the large intestine, resistant starch undergoes microbial fermentation, producing beneficial short-chain fatty acids like butyrate that support metabolic and gut barrier health. Because this retrograded structure is relatively heat-stable, gentle reheating generally preserves much of the resistant starch formed during cooling, though the overall glycemic effect varies according to the specific food, cooling duration, and individual digestive response. The order in which you eat different components of a meal can meaningfully influence postprandial glucose dynamics. In randomized crossover trials, researchers have investigated the metabolic outcomes of consuming vegetables and protein before carbohydrate-rich foods compared to eating the same meal in reverse order. Human evidence demonstrates that starting a meal with non-starchy vegetables, rich in dietary fiber, alongside lean protein and healthy fats, can help moderate subsequent blood glucose excursions. This effect is attributed to several complementary physiological mechanisms, including a slower rate of gastric emptying and the enhanced release of satiety and glucose-regulating incretin hormones, such as glucagon-like peptide 1 and peptide YY. Rather than eliminating dietary starches, simply structuring your plate so that a colorful salad, steamed vegetables, or a protein source precedes your rice or potatoes can support a more gradual metabolic response. Introducing dietary acidity into a meal represents another evidence-based approach to modulating carbohydrate digestion. Clinical studies suggest that consuming vinegar or other acidic foods with a carbohydrate-rich meal can modestly reduce the subsequent postprandial glucose response in some people, likely through a mild delay in gastric emptying and temporary modulation of digestive enzyme activity. A complementary principle applies to traditional sourdough bread. Unlike wrapped commercial yeast loaves, authentic sourdough undergoes extended fermentation with wild lactobacilli and yeasts. This fermentation process produces lactic and acetic acids and modifies the dough's gluten and starch matrix, which can produce a lower glycemic response than some conventional breads, although the effect varies substantially by recipe, fermentation time, and individual physiology. Finally, physical movement after eating plays a crucial role in post-meal glucose regulation. After eating, blood glucose typically rises over the following period, with the timing and magnitude of the peak varying according to the meal and individual metabolism. Engaging in light, non-strenuous physical activity, such as a 10 to 15-minute gentle walk or light household movement within this post-meal window, triggers skeletal muscle contractions that stimulate non-insulin-dependent glucose uptake. This process promotes the translocation of glucose transporter type 4, known as GLUT4, to muscle cell membranes via energy-sensing cellular pathways, drawing glucose directly from circulation without requiring additional insulin.
By thoughtfully combining starch cooling, intentional meal sequencing, dietary acidity, and light post-meal movement, you can comfortably enjoy traditional carbohydrate staples while supporting metabolic health. Medical disclaimer: this video is for educational and informational purposes only. It does not constitute medical advice, diagnosis, or personalized treatment. Always consult a qualified healthcare professional before making significant changes to your diet or lifestyle.